Fantastic. Okay, a very good morning to you, bright and early on this Thursday. Um, I'm just going to wait a few moments because it can sometimes take a few minutes for people to log in via Zoom. So first things first, if you can see the chat function on your screen there, if you've joined us, could you just say hello or hi or something in the chat, just so that I know that you can hear us okay and we're coming through loud and clear and that the chat works. So you could just say hi. Brilliant. Thank you very much. That's really, really helpful. Good morning to everybody. Hello, hi. So whilst we're waiting for people to log in, waiting for people the attendees to start to join us, just for a bit of fun, just purely out of interest, if you could let us know in the chat where in the world you're joining from. We have these sessions scheduled at different times throughout the day. So we had one on Tuesday that was later in the day. We've got some that are going to take place at lunchtime, and these ones are for us in the UK early in the day. They're at the start. So if you could just pop in the chat and let us know where you're joining from, that'd be great. So interesting, yes. So we've got someone joining from London, also India, Bangkok, and someone from Germany. And that does reflect what we found on the first one on Monday. We have people from Thailand, Singapore, Indonesia. So it's really interesting to know that these sessions are very good for people at that time and point in the world, and it does actually work. But just to let you know as well, one of the first things to say is this session is being recorded. All of our webinars are being recorded, and there will be links shared afterwards, so you can watch back this one and all the others as well. So if you missed the one on Monday and Tuesday, and if you're not going to be able to make the ones that are coming up tomorrow and on Monday, you'll be able to watch them back. But of course, being here in person, you get to participate in them, which is particularly exciting. So my name is Kim Morgan. I'm the Outreach and Communications Coordinator at UCL Biochemical Engineering, and it's fantastic to have you with us. First things first, I just want to say that in this webinar, which will be presented by Joan, and also colleague Steffi will be attending as well, but the presentation will be by Joan. If anything comes up scientifically or in engineering terms that you don't understand, don't let that concern you. If you were to come do a degree with us, you would be taken up to that level. That's what we do here, is we raise you up to that level. And in fact, speaking about that, what I'm going to do is I'm going to launch a couple of polls, because I'd really like to know a little bit about the composition of the audience. It's really helpful for us to know what sort of level you would be thinking of. So are you a prospective student? Are you considering being an undergraduate or postgraduate, or is that not applicable to you? So if you could let us know in that, it'll help us. It just really helps us. Different webinars that we've done have had different compositions of people. So the one we did on Tuesday with stem cell and gene therapy was slightly different to the ones we've done on vaccine manufacture. And it's very interesting for us to know why. So in terms of the composition for this one, it seems that the vast majority of people are more likely to be doing or considering doing an undergraduate degree. So like BSc, BEng or an MEng. Fantastic. That's very, very helpful indeed. And it also helps us gauge this session as well. Okay. And the other thing we just want to know is, do you have an offer? So if you've got an offer to study, can you let us know? This is we promoted this webinar through UCAS emails and through internal systems, but we're really interested to know whether some of the people who have an offer to study with us or somewhere else at UCL are joining this. And it seems that most of the people who are joining don't have an offer, which is very, very interesting. But some do. So we've had a couple who do have an offer to study with our department. So that, again, it's very helpful for us to know that. If you've got an offer, fantastic that you come to join us today, and I hope to see you at one of our offer holder events. But in the meantime, thanks for coming along today. And if you don't have an offer, we hope this might push you to apply to us and come and study with us. Okay, that's fantastic. Thank you very much. Really appreciate that. So what I'm going to do now is I'm just about to hand over to Joan. If you have any technical problems, you'll see that on the bottom of the screen, whatever device you're on, you've got different ways you can chat to us, so you can reconfigure the screen, but you've also got a Q&A function. If you have any technical problems, please use the chat. So if you can't hear us properly, it's a problem with the slides, anything like that, let us know in the chat. Myself, Joan and Steffi will see that in the chat. We're really looking forward to your questions. We can't wait to take them. That's why Steffi's here. Steffi is one of our senior academics in the department and knows and is really looking forward to taking those. But please put in the Q&A function. It's really helpful for us to be able to answer them. If you put your questions in the chat, we can sometimes miss them because they sort of disappear, whereas the Q&A function means that they're really much easier to follow. You can also post them anonymously if you want to. So I'm going to hand over to my colleague Joan. Joan is going to start the talk. At the end, we'll come to your questions. But if I turn my camera off, I will be here in the background. Joan, over to you.
Sure. Um, Thank you for introducing me. And thank you again for joining this session in this morning. My name is Joan, and then today we'll talk about like how can you accelerate vaccine manufacturing without cells. Um, 1 second. Yeah. So this is our content for today. First, I will tell you a little bit about myself and how I ended up doing what I'm doing now and why proteins matter. To spoil you is because it's the main ingredient of the vaccines mostly, and then how we currently manufacture them. And I will introduce some new technology called cell-free system. And then at the last, we'll have some Q&A session, as Kim mentioned. So just a little bit about me. I always have think science is kind of cool, and then was been fascinated by how it can improve the world, and that's why I chose my bachelor's degree in chemical and biological engineering, which I did back in South Korea. And then after that, I continued my master's in similar degree, chemical biotechnology, but in Munich, Germany. And then I also had some work experience in a biotech startup there. To talk a little bit about that experience in Germany, I got fascinated by the technology using synthetic biology, which is something that you use modify biological systems to produce something useful. And back then, my focus was more sustainability. So I was using some cells to produce useful materials that can substitute petroleum-based chemicals. And that technology will be explained further later on, but this new technology really fascinated me again, and then I wanted to study more about it. And then that's how I ended up applying for my current position, which is called NHD, is like a PhD with an industry collaboration. And now the focus has changed using the same technology, but producing some materials that's useful for public health, like medicines and therapeutics. So throughout my journey, I was generally just interested in applying biotechnology for a better world. And I will, go directly into this technology that I'm keep referring to. But before that, I want to bring up that facts about the vaccines. I'm pretty sure that you all know about vaccine, and everyone in this session has had at least one vaccination in their lives. It can be a flu jab, it can be a COVID vaccine recently, and people who are raised in UK or somewhere else around the world must have had some sort of childhood vaccination plan. And when you look at the numbers, it is really staggering. Like 19, I just brought examples from England, UK. Flu jab in 19 million in England only, which means like 1 in 3 only last year. And COVID worldwide has been accumulatively 14 billion, which means 70% of worldwide population has had one vaccine at least. So vaccines are absolutely everywhere, but here's the question. What's actually inside them to make that work? The answer is obviously protein. So there are many types of vaccines, like is shown in this graph, but over 50% of all of them are protein based. So the main ingredients that activates your immune system in vaccines are or protein. But it's not just vaccine. It's also for any modern days medicines are mainly based on proteins. For example, to treat the cancer, autoimmune diseases, Alzheimer's, monoclonal antibodies are used, which is a protein. And also, maybe you have heard of it recently a lot. GLP-1 analogs for weight management is also protein-based. And not only that, there are many, many diseases that are treated by protein-based medicines. So the demand for protein-based medicines and vaccines are very growing and huge. And this raises the question how these proteins are made. And this nature's law applies for how proteins is made. So every protein in your body is built from the instructions coded in DNA. And if we want to make a specific protein, any vaccine protein, then we just need to give a cell the right DNA instructions, and cell does the rest. So cells takes this DNA instruction and then change it to RNA and then make it into protein once we give the right instructions. So right now, this becomes the fundamental of every biomanufacturing. So the way we make proteins using the living cells currently, so we reverse the nature's law first. So once scientists identify which proteins are needed to be synthesized, then they can synthesize the DNA that instructs this protein synthesis. And then we take cells that could be bacteria, yeast, mammalian cells, or insect cells even. And then we give these cells this instruction DNA. And we collect the cell that has the right instruction inside them and then grow them in a system that's called bioreactor, controlling temperature, pH, or nutrients that cells need. And then under the right condition, the cells will grow and produce a protein. And then once the cells are grown and produce the proteins, we need to collect them and tear them down to purify the protein. And this becomes the core ingredient for the medicine or vaccines. But the problem here is that the entire processes take too much time. So even just selecting the right cells takes months, and then making them grow and happily grow in the big bioreactors takes other months. So when you think about the COVID and then thinking about this whole entire process just taking months and months to set up the process, then it can be a really bottleneck. And also, if you think about the patients with a rare disease who need urgent treatment, then this entire process can be very excessive. So that's where this idea comes. What if we remove the cells and streamline those processes? So the scientists came to think of, what if we just use what's inside the cells, just take necessary materials from the cells and use those machineries to make the protein. And how it works is we tear down the cells and take only these inner materials and store them either in the freezer or freeze dry them in the storage. And whenever we need to produce new protein, then we just synthesize the DNA that gives this instruction and takes this extract from the store and then mix them, and then protein is made. And this is called the cell-free system because it doesn't rely on the living cells. And the main advantage would be obviously the speed. So once you identify the protein that's needed, you make this DNA and take the extracts from the store, and then protein is produced. And then since it is already outside the cell, you don't need to worry about much purifying them. And you don't also need to worry about big bioreactors trying to make the cells happy. And it makes purification also simpler. And then this mixing the cellular extracts with DNA takes only minutes. And this production of protein stage takes only hours. So when you compare it to current cell-based methods, which takes months and months, it is really faster. So this is like general overview of the system, and I want to talk about a little bit what other scientists do with this new technology called cell-free system. And before that, there are also other advantages of cell-free system. As I said, it is faster, but also the system itself can be freeze-dried and stored easily in room temperature without the need of like excessive cold chain. So you can just bring the cellular extracts anywhere in freeze-dried format and make them produce proteins that's needed. And some scientists have even taken freeze-dried cell-free system into the International Space Station. They brought them on a mission this dried cell-free system and then rehydrated them and successfully made some fluorescent proteins up in the orbit, which I think is very cool. So what you're seeing here is this fluorescent protein being produced in this dried cell-free system in the space. So I imagine doing the same experiment using the living cells will be really, really difficult because you have to keep them alive the whole trip. And also other advantages of cell-free system is that it can basically work at any scale from tiny droplets to huge bioreactor scale. So being able to produce protein in these tiny droplet scales make it possible to test many, many proteins at once. So what you're seeing here is the machine commercial device that helps scientists automatically test different conditions to make proteins using cell-free system. And also with the rise of AI, scientists now have many, many potential drug designs to check. And cell-free systems is a fast, flexible platform to test all those conditions. And finally, some companies are actually using the cell-free system to make medicines now. The example here is the factory design photo that Sutra Biopharma is using cell-free to produce some medicines, monoclonal antibody-based cancer treatment medicines here. And there is also another company called Lenio Bio, which I am collaborating with, which is from Germany. And my research is to develop a platform using cell-free to manufacture vaccines as well, but I use plant system. So common cell-free host cells that are being used are usually bacteria because they're easy to grow and yeah, they're well known. But I use plants specifically because many vaccine and therapeutics proteins need some complex proteins, like proteins need to be folded in a specific way to be active. And these things bacteria cannot usually do, but plants can. So I use plant cell-free system for vaccine manufacturing. But just to point out, I'm not actually growing the green plant. I grow like special type of plant cell that can grow in the reactor, like when you imagine a beer, the yeast grows in the liquid. So that's what I'm growing. And then I take the cellular extract from those plant cell and mix it with the gene, and then the protein is produced within some hours. So the cell-free system is very exciting, but they are very new technology. So there are still some challenges need to be tackled. The main ingredient of the cell-free system is obviously the cellular extracts, the machineries inside the cell. So what I do is take a plant cell and then disrupt them. Then it will burst out many, many small molecules inside that extract, that cell. Some are known, but many of them are unknown. So I don't actually know which one is really helpful for protein producing or which one is getting in the way. So right now, what we need to do is to understand what is actually happening inside the system. And secondly, we need to boost the system to produce more protein. Currently, cell-free systems don't make as much protein as cell-based methods. So if you want this technology to be really adopted, then we should improve the protein production capabilities. And that's exactly what my PhD is about. So the first part is about understanding the system. So I've been developing new methods to observe what's happening inside my plant cell-free system. So the graph here shows one of the results from my methodology where I could track how much proteins are made along the cell-free reaction. So here what you're seeing is how much protein A and protein B is being made along 24 hours. And understanding how my system behaves helped me figure out what to tweak in my system to make it better. And based on that, I am also developing some methods that can make my system to produce more proteins. So I just turned it as a finding secret recipes to increase the protein production because it's really like a cooking where you have to find the like optimal temperature and then try some new ingredients to find the recipes that works well. So the graph here shows how I am using my own developed recipes to improve the protein production for different protein A and protein B. So control method here shows how it produces protein, how much protein A and B are made using this normal method that my company uses. But this new recipe one that I developed has doubled the protein A's yield, how much protein A is made, and also it improved protein B's production up to 70%. Recipe two hasn't worked very well, but this work is still ongoing, so I'm trying to find out which method works best. And I'm pretty optimistic about it, like to push this even further. And this improvement really brings us closer to making cell-free system actual reality of manufacturing real medicines. So that was the brief overview of what I'm doing and why I'm doing that. And this is my final slide. And before closing up the talk, I want to leave you some glimpse of the future with this exciting technology, where this cell-free system can lead us. So we can imagine something like pandemic preparedness. As I said, cell-free system streamlines the vaccine manufacturing process a lot. So once you have a new pandemic, then you can design a vaccine in days and then manufacture in weeks, not years or months as it used to take. Also, as I mentioned briefly, cell-free system doesn't need massive bioreactors, and also it can be stored freeze-dried and can be carried around easily. So it will be really helpful for those developing countries who doesn't have a lot of resources to grow the cells and produce proteins. And lastly, it works in a small scale very well. So it will be helpful for personalized medicine in the future, or whenever a person has a specific type of tumor or some rare cancer, then it can be used to make small batches of custom proteins for those individual patients, which will be very difficult with current cell-based methods because it will be very expensive. Yeah, so before I really finish, I want to quickly thank people that made this research possible. So I'm incredibly thankful for my supervisor, who is also here, Stephanie Frank. And also I have other academic and the industrial supervisor, and this is our academic group. And also the funders who supports my research. Yeah, so I think we can go into some Q&A session. Hope you enjoyed the talk. Hello.
Fantastic. Thank you very much, Joan. That was great. I'm
Should
seeing
I
we've already got a
stop
question.
sharing?
No, that's that. Well, yeah, yep. That's stop sharing. That's great. Thank you very much. That was great. I can see we've got one question that's coming already. Before we go to that, though, I'll let Joan and Steffi read that question. But just want to say, it's a really good one to start off with. Yeah, so thank you very much for joining. We're looking forward to your questions. Somebody's already seen that you've got the Q&A function. Please put in the questions in the Q&A box. It really helps us to answer them. If you've not signed up for the sessions that took place early this week, or the ones that are coming up, I will send a link out to everyone who's registered to be able to watch them all back, like you'll be able to watch this one back. But there is still place available for tomorrow's one on lab-grown food, lab-grown meat specifically. And we've got a session on Monday with Matthew Banner, who's going to be talking about automation and artificial intelligence in the biopharma sector. We did a session earlier this week, and there were a lot of questions about artificial intelligence, so he's going to really do a deep dive into that. Matthew also works, he does something similar to Joan in that he's working with us, but also working with a company as well. So if you've got any questions about what it's like to work between the interface between academia and in the industry, please come along to that, or please ask questions of Joan now. Joan is very well placed to talk about what it's like being in the sector. What I'm going to do is hand over to Joan. Joan, if you'd like to read out the question, and I think it's the questions that are coming in. I'll leave that with you.
Yeah, sure. I've got a quite technical question. How does the use of cell-free system affect the affordability of vaccines for people compared to the older technology that requires the use of cells and bioreactors? So as I mentioned, this it makes the system quite accessible and affordable for the individual who needs personalized medicine care. So still now, big medicines that are treated for cancers like monoclonal antibodies, in that term, the cell-based methods is more affordable. But when it comes to more personalized and small batches, I think the cell-free system can be more economical options.
Just to
Yeah,
extend on that, before Steffi comes in, just say this is something we've also covered in Rana's session on Tuesday. So you will be able to do a deep dive into this again. But Steffi, over to you.
yeah, thank you, and nice to meet you all. I'm Stephanie Frank, an associate professor in engineering biology in the biochemical engineering department, and I'm Joanne's PhD supervisor. This is a really good question you were asking about manufacturing, and there are a lot of efforts into trying to kind of understand what are the costs of these different technologies. Um, Going back, so childhood vaccinations, as you rightly said, they have been developed many years ago, tens, 20, 30, 40 or more years ago. And they are based on kind of, these kind of first technologies, but they have been developed over such a long time that they are quite cheap now. So we still use a lot of the original techniques to make a certain childhood vaccinations whereby cells are still grown in bioreactors. We take parts of cells, bacteria, or we make viruses and take parts of those and after they get inactivated and so on. And these techniques are relatively cheap compared to maybe some of the more recently developed techniques. But the more recently developed techniques, such as the ones that Joan was talking about, or say mRNA vaccines and so on, they're really catching up and they have so many advantages and they're very modular. We can design and redesign quickly, particularly like Joanne mentioned this several times in the situation of a pandemic. These kind of technologies, they become really important and they can be really helpful in addressing, for instance, pandemic outbreaks very quickly, as we've already seen during the COVID-19 pandemic. So we ultimately will see them becoming more comparable in terms of costs and so on. But the speed is important, and John mentioned that. And then also the storage, we shouldn't forget about the need for the cold chain. So when we transport vaccines, often they need to be kept at very low temperatures, -80, -20. And with these kind of cell-free systems, you can potentially transport them at room temperature, which makes, it's very advantageous, particularly in regions where we don't have that stringent cold chain. So I just wanted to add these kind of aspects as well. Okay, Joanne, I think the next one
Yeah.
is for you.
Yes. So I have this question, how I got on this pathway, which kind of courses I did, and if I found anything limiting me from getting to this point. And yeah, regarding that, so as I introduce myself, my degrees have been heavily biotechnology focused and engineering focused. But during that, I tried to get as many as hands-on experiences because those things were quite limited from where I did my degree. But I think here you get to get those hands-on experiences in the lab or presentation skills during your course when I see the UCL when I teach the UCL students here. But yeah, and so I tried to expose myself to a lot of those industry opportunities. Even for my master's thesis, I did it in the biotech company, which really developed where I really developed a lot of skills sets. And also doing currently my PhD with the industry, I get to expose to the company environment as well, which really helps me proceeding with the research and also just generally my soft skill sets. Yeah. Yeah. So yeah,
Yeah.
that'll be my advice.
Thanks, Joan. I wanted to just add that, like Joan's journey, everyone's journey might be slightly different into coming into the department. And as an undergraduate student, because I've seen the survey that Kim did at the beginning, and I've seen that a lot of you are interested perhaps in doing an undergrad degree. As an undergraduate student, you would also be exposed to kind of industry input into the degree design, but also like feedback from industry partners. So the department historically has worked for many, many years with industry, biotech industry, biopharma, and a lot of our graduates go afterwards into industry, and they come back. Some of them come back later and they help us on the courses. They give feedback to the students. They advise students on certain coursework. So we have that connection, and I think that's quite unique to our department, and is something that we really value. These close connections to many, many different companies. I think around 100 or so companies are have some connection with the department.
I just want to jump
Yeah.
in because we've had another question come up, which is interesting with Joan. But I just want to jump in and say we've blocked out until 10:30. Please keep your questions coming. If we come to the end of it, we're happy to wrap up the session when we've answered all your questions. But we've made this time available, so it's absolutely open to you. So two really good questions coming for Joan there, and they're very specific to you about your experience. So I'm going to
Yeah.
hand over to you for those.
Yes. I'm really happy to see these questions being asked. Yeah. But so the first one, someone asked me about my work experience in Germany, what I worked on, and how that experience was. So I was working as a scientist in a German biotech startup. And what I've worked on was using this cell-free technology, but to produce proteins that are helpful for environment, not for public health. So I was using similar technology. But just the target product was different. I was making system to be able to produce petroleum-based chemicals, but not using the petroleum, but just using the cells and feeding them some sustainable like waste as some waste so that we don't have to rely on petroleum heavily. And that experience was really eye-opening because I got to expose to this new technology working in a startup. And also, it was my first work experience, which so it really helped me like maturing as a person as well, and got exposed to like new culture with different scientists from people around the world. Yeah. I really enjoyed it. I just wanted to study more about that. That's why I came here now and working with different company. Yeah, and to answer, it leads to me to this another question, like how I made the switch between a focus on sustainability to vaccine related public health. So as I mentioned, I was using the same technology, cell-free protein synthesis, cell-free system. But as I said, just the product was different. You can use this technology to make all different types of proteins, and some proteins would be helpful for environment, and some proteins would be used to medicines and therapeutics. Yeah.
Brilliant. Actually, Steffi, I want to just ask you a little bit because people are talking about roots in experience. First of all, just to explain what the pilot plant is that we have in our department, UCL Biochemical Engineering, and then perhaps mention iGEM because I think not enough people know about that.
Yes. So Kim just mentioned the pilot plant. So we are a department with, in another sense, very unique because we have a what we call a pilot plant, which is essentially a, a big room with equipment that allows us to look at different steps of manufacturing a product. And that could be a vaccine, it could be some kind of genomic medicine, or it could be an antibody. Or another product, could also be artificial meat or things like this. We have lots of different applications, and our department is very focused on researching and doing novel research and kind of developing manufacturing processes to make these kind of products. So the pilot plant is something that we use also with the students. They go and see the equipment, they get hands-on experience on the equipment, and they learn, like, for example, how do you break the cells open to get the product out? How do you then purify the product so you remove all the components from the cell that are basically not needed and not wanted? How do you then formulate your product? So what you have to add to the product and so on. So the pilot plant is almost like a small version of a kind of commercial manufacturing process where you work with maybe tens of liters, maybe even a little bit higher, up to 100 liter of, let's say, a big culture of cells. So as we call it, cell culture, where we grow the cells, and then obviously doing all of these steps to the final product. And it really allows you to research these steps because, it's between the lab, basically what we call the wet lab work, kind of fundamental research work that we do as scientists, between that research and the manufacturers, like let's say the biopharma industry obviously works on much, much larger scale. They grow something at thousands of liters. So students really get to see that equipment, and that's very unique, as I said, to our department, I would say. The other point that you made, Kim, was about, so as you have seen from Joanne's presentation, she mentioned synthetic biology a few times, and you have most likely heard of this terminology. So synthetic biology has experienced a kind of a lot of attention over the last 10 to 20 years with a lot of developments in this field where we can re-engineer biology for applications like vaccines, for example. And essentially, we enable our students as much as we can to work on independent synthetic biology research, where they are designing some DNA and they're making some products and they work on a real world problem. And this competition is called iGEM. Some of you might have heard it or maybe have taken part into this competition in your high schools. It's the biggest worldwide synthetic biology competition. At UCL, we have been consistently, historically, able to enter the competition, and we are looking to keep doing that in the future. So students that come to us can apply for the competition, and if they are successful, they can work on this kind of real-world synthetic biology project as well. Okay.
Sorry, I just want to jump in
Answer the
because
question, Kim.
so there two questions have come in, but one of them is a more general thing about coming to university or coming to biochemical engineering as an A-level IBAC student. But Joan, there's a question that's come in about if I'll read it out. It's okay. Have you considered the integration of epigenetic modifications to accelerate protein production and help with the protein stability? It's I think that's very much over to you, Joan.
Yes. I'm really surprised by I assume the student is preparing for PhD. I'm not sure. Yes. To accelerate the protein production, so my approach is to modify this generic cell-free extract itself, not directly like DNA related or nucleic acid epigenetic modification. So but you can also consider that to accelerate the protein production and help protein stability. But it's just my approach is different and my focus is a bit different. So I'm working towards more making metabolism of the cell-free extracts a bit different so that it is more optimized in that way. Yeah. So that would be my answer. But I think it is a very good point that you can also consider to make system better. Yeah.
Thank you. Um,
Okay.
so the other question that's come in, I think it's I'm gonna address this more at Steffi because Steffi sees a lot of undergraduates coming in to us. So they only recommended science related or lab experiences, supercurriculars for a student doing their A levels before they come to university to do a degree course.
Mhm.
It is a question that comes up: how can I prepare to go to university? And there's kind of two strands to that. I'll come in a bit afterwards and talk about your UCAS statement if that's something. But in terms of preparing you for university, Steffi and then Joan, what would you say?
Yeah. Okay. Thank you, Kim. So I think that's a really good question. And I think the key is perhaps not so much how much experience and so on. I think it's really your interest. Are you, like, say, for a biochemical engineering course, it's really useful to have done a little bit of reading. What are the different programs? We have a number of different programs that maybe at this point is worth mentioning. So we have the biochemical engineering undergrad degree called BEng, so it's 3 years bachelor's program, and then we have also the master's level. So that's a 4 years program also in biochemical engineering. And so these will be courses that where students really learn also the underlying engineering principles, for example, for manufacturing processes that we briefly discussed earlier. But that it's essentially these research programs are really covering everything from molecular design all the way through to different steps of manufacturing, all the way through to, is this a feasible process, as someone has earlier asked? Can we calculate that? So it's really, are you interested in this? Are you interested in making molecules for these applications that we discussed? And then there's certain, obviously, entry requirements that you can find on the website for the different courses. So, maths or physics or chemistry useful for these kind of more engineering focused course programs. But we also have the BSc in manufacturing of new medicines, which is a very long name, I know, but it's quite, so that also would kind of want you to have a bit more of a biology background, perhaps. And it's really more focused on the different types of products that you can make, like genomic medicines, like maybe, DNA medicines, viral vectors, vaccines. And so having a bit of a biology interest is very much advantageous. You don't necessarily have to have a lot of practical experience because you do learn in your first year in the teaching labs in UCL East how to clone DNA, how to make a protein, all the things that we just talked about. You will learn those things, and then you get a bit more hands-on experience on using equipment in a pilot plant, as we said earlier. So, these are part of it. And I should also mention that many of our students are very proactive to secure like studentships when they're in the program. So typically in your second year, you'd be looking to go and work maybe within academia or with another research lab, or you work in industry, or do something else, work with another kind of industry partner to gain a little bit more experience. So I would say not to worry too much about having all of those skills, because that's where they come into the department to get training. Okay, Kim, I give back to you.
Thank you, Joan. I mean, you're fairly close to this. I mean, you're doing a PhD. So, is there anything that you would advise a potential undergraduate that maybe you did that was useful or that you wish you'd done?
Um, I just generally agree with Steffi. You don't really need like a practical experience before you come into the university. Those are the things that you gain during your degree here in the university. So I wouldn't necessarily worry about it, yeah, that much.
Brilliant. I mean, it's one of the things which, because we've done some webinars with students talking just before induction, so they've been answering questions of people who are about to come in, and it covers a wide thing. One of our excellent students who did the undergraduate, did an MSc, and now doing a PhD, Tamas Laufer, he said that, you know, for him, taking time off before going to university was really valuable. You know, taking that summer, spending time with friends, because he said when you come to a degree, it is intensive. You know, it's there's a lot of hard work. He said it's very rewarding, but it's good to come into that well prepared and rested. However, some people said if you want to come, if it make you feel better for coming in prepared, read up on what's going on in the sector. Read some papers. This is just from students who said they, you know, they enjoyed preparing themselves and thinking what kind of things are going on in the sector, what academics are working on. In general terms, you know, what are the exciting things that are going to happen. So that's something. And also with your UCAS statement, it's a question that comes up because we're the only department of biochemical engineering. People who apply to do degrees with us, they can't be really singularly focused on biochemical engineering. It's going to be biochemical engineering with other subjects, or possibly medicine, or, you know, other subjects. So what we hope to see in the UCAS statement is that someone is interested in using biology and chemistry and solving the world's problems, and, you know, the kind of things that we're looking to solve, that they have an interest in that, and express some of it in the statement. But we understand if you're focused on other things as well. So, you know, we do really understand that. Okay, I think we've answered all the questions that. Sorry, somebody did actually post a message asking about PhDs. I've given you my email address. Feel free to reach out to me directly. You ought to have got my email address through communications, and I will get in touch with you afterwards to let you have a link to the recording. If you say you're interested in doing a postgraduate talk program or our MSCs or our MRes, or if you want to do a PhD, I can put you in touch with the appropriate team in our department and we can take that further. Otherwise, I want to say a huge thank you to Joan for the presentation. That was great. And thank you, Steffi, for joining. Really appreciate your taking the time to join us today. But most of all, thank you to the people who came along today. Thank you for attending, and thank you for your questions. Some of them, you know, they were really good questions. Some were very technical and demanding. And thank you for your kind words. It's great that some of you that you joined us, and I hope to see some of you again, and I hope to see some of you in person at UCL in the future. So have a great rest of the day, and thank you for joining us.
Thank you, everybody. Have
Thank
a good
you.
day. Bye.
Bye.